we start with cell division.
What chapter?
Chapter 9, page 154.
Turn off the lights. Should we?
Yeah, we probably should. Maybe, could you pull that blind open on the left side there? To get a little more light in here.
Nothing for you.
No soup for you.
No soup for you.
Nothing for you.
What's that from? That's Seinfeld. Seinfeld's a little old for y'all, isn't it? It's still on. Okay, so this is a picture of a cell dividing into two cells. Do you remember? Did we ever talk about why cells divide?
Yeah. There's something about like the fibers go to the outside and then push out.
Yeah, but why just, how come, why not just not divide? I thought we talked about this.
They need to make more sense.
Why would you need to make more cells?
Because the other ones die.
Yeah.
Oh, to evolve.
Yeah, that helps. That's a good reason too. Your body's made of 80 trillion cells. Did you know that? 80 trillion cells. That's a lot. Why not? Why wouldn't it be made of 80 million cells? That's
Surface area.
That's still a lot, and that's a million times less than 80 trillion. Somebody said it?
Surface area.
Surface area.
Or something ratio.
The surface area to volume ratio. Do you remember that? That's why cells really divide. It's because when they grow and get bigger and bigger, they suddenly don't have enough surface area to feed all of their volume. So did we talk about that?
Yes.
Okay, good. And so what they'll do is they'll split. So now they have enough surface area. And so cells never get bigger than a certain size. And so what we're going to study this chapter is how the cells divide. And it's through what we call the cell cycle. And the cell cycle consists of a long phase called interphase that has three parts to it called G1, S, and G2. We're going to go over all this today. And that's kind of, interphase is kind of normal cell life. Normal cell, the cell is just going through its life. You can't see anything abnormal going on in interphase if you're looking at a cell under a normal microscope. But then all of a sudden you hit this M phase. M is short for mitosis. And mitosis is the division of the nucleus of the cell. The nucleus of the cell will go from a single nucleus in the cell to two nuclei in the cell. And this division of the nucleus we call mitosis. And it consists of 5 phases: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis.
So mitosis is the separation or the dividing of the nucleus?
The dividing of the nucleus. And as soon as the nucleus is divided, then cytokinesis happens, which is the division of the cell itself in two. Which is a pretty quick process when the cell just kind of splits in two. So this is what we're going over today, the cell cycle. And so cells, after they split in two, start again in this what we call G1 phase. G1 phase is a growth phase. And it's when a cell is just growing and getting bigger, doing its normal life, eating, respiring. Some cells do photosynthesis, making the things they need to make. And that's the G1 phase. some cells will stop themselves in the G1 phase. There's a little stop point. Let me go forward in here and see if it shows the stop points. Yeah, here we go. There's a little stop. See that stop sign? That means a cell can stop itself right there. And some cells grow and then stop growing, and they're just stuck there for the rest of their lives, and they never reproduce anymore. Have you ever heard that your brain cells don't regrow? And once you lose a brain cell, you'll never get it back? That's, for the most part, true. There are some brain cells that do multiply, but many of them do not. So they'll grow and get a certain size, and then they're done. No more reproduction for them.
Why not?
There's really no need for it. They can live for many, many years and do the job that they do, and you don't really need them to multiply anymore. There's really, in the brain, there's no room for them to multiply anymore. So they stop multiplying.
Do they still function?
Oh yeah, they'll function for your whole life. But you go drinking a lot, or ram your head against the wall, or whatever you guys do on weekends, and the cell dies, then you're one less cell. Now luckily you have billions of cells, but by the time you get 90 years old and you've done a whole lot of drinking over your whole career in life, then you've lost a lot, and you become a little bit senile. You don't function as well as you used to. So that's what you have to look forward to. It's all downhill from here, y'all. Anyway, what was I saying? So you go through, cells go through G1, and then cells go through this S phase. And S, if the cell is going to further divide, they'll go through an S phase where they copy their DNA. And every cell has DNA in its nucleus. Y'all remember DNA? The model Jonathan made for us here? DNA is, deoxyribonucleic acid. It's the information that the cell carries. And it's a really long strand. This is just a short piece of it. It actually goes real long, and I like to describe it as fishing line. Imagine taking a whole reel of fishing line and undoing it. You'd have this real long strand, right? And it's all spread out in the nucleus. And in the nucleus of a cell, there's actually 46 long strands of DNA line in the nucleus of a human cell. It's cut into 46, the DNA is cut into 46 pieces. And in the S phase here, the DNA is copying itself. And so each strand of DNA, each long thing like fishing line, copies itself. So there's two of them. So you'll go from 46 long strands to suddenly 92 long strands during the S phase of interphase. This is in a human cell, there's 46. Every organism has their own number of what we call chromosomes, which are long strands of DNA. Have y'all heard that humans have 46 chromosomes in each cell? Yes. That's somewhat common knowledge. Not everybody knows that. But... Oh no! Jonathan. Knocked one off. We'll have to glue that back on. Good model, by the way.
Very stable DNA.
Little mutation there, my guy. So after the S phase, where the DNA has been copied, then you go to this G2 phase, and the G2 phase is some final preparations for cell division. So there's some proteins that have to be made. There's some organelles that have to copy themselves, like the centrioles. Do you remember the centrioles in an animal cell? They'll have to copy themselves, and then there's another stop sign here, a checkpoint, where everything stops and the cell kind of examines itself and makes sure that it has everything it needs to go through this mitosis. cell will start dividing actively, and that's the mitosis phase. Prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis.
Do we know all of that?
Yeah, I'm going to go over them in about 2 minutes here. There's another checkpoint during mitosis where the cell checks itself. And what it is special enzymes locking on to the molecules that need to be created here. And if those enzymes do not lock on to the particular molecules, the cell realizes there's a problem. And you know what the cell will often do if it comes to problems here or here?
Kill itself.
It'll kill itself. Cell suicide. It's called apoptosis. The cell can actually kill itself. Isn't that cool? You know how it kills itself? It doesn't stab itself with a knife. Doesn't go in the garage. I could think of a lot of ways to kill yourself. It doesn't do anything like people do. You know what it does?
What?
Think about it.
It eats itself with those like lysosomes. They open up or something.
Yeah, it creates a bunch of lysosomes and opens up the lysosomes on itself and digests itself. Isn't that interesting?
Wait, so the cell needs to hook on to the enzymes?
No, there are enzymes inside the cell that are basically looking for the proteins that need to be made. And if they don't find those proteins, then they'll initiate, there will be like a chain reaction that initiates the cell suicide process.
So why does the cell need to kill itself?
Well, if something's wrong, then the cell might copy itself incorrectly. You might end up with a cell that has a mutation of some sort. You could end up with a cell that won't stop dividing, for instance, that just divides over and over and over. That's called cancer. And one bad cell that goes cancerous can kill the whole organism. So, to keep that from happening, to try to keep that from happening, a lot of cells end up killing themselves. Yes?
What's it called when the cell kills itself?
Apoptosis.
How does apoptosis fail when you get cancer?
Well, what's happening is those enzymes that are supposed to be checking things, one thing that can happen, the checkpoint enzymes, they can get screwed up and mutated. And so they don't initiate the apoptosis process correctly. And then the cell doesn't get killed. And if it's a cell where its mutation was it keeps multiplying and nothing's killing it, then you got cancer.
Can you watch a cell kill itself?
I actually have some video of it here somewhere. Let's see if I have some here. Oh, well, here's pictures of it. This is what it looks like when it dies. It forms these little blebs and fragments into a bunch of little pieces.
Why not little pieces?
They're eaten up by other white blood cells that come along and clean up mess. Let's see what this is. I think this might be a pretty lame video. Yeah, this is a lame video. It's a normal cell. That's an apoptotic cell. So it just kind of comes in fragments into a bunch of pieces.
Does it happen that fast?
No. It talks about it on the top right of page 155. So, as far as interphase goes, let's go back to this. As far as interphase goes, there's G1 when the cell's getting bigger. There's S when the cell's copying its DNA. And then there's G2 when the cell is getting ready for cell division. These down here are a bunch of videos just telling you that, and they really, I need to get them off of there because they're not exciting, and it's just a guy talking. He's just a little bit more boring than I am, which you can imagine it's terrible. I'm not going to show all this. And so then we start the phase of mitosis. Prophase is the first phase of mitosis. Now the cell's going to divide. And what we see, if we're looking at a cell, is we see the DNA, all those long strands, we see them coil up into what we call chromosomes. The strands wind themselves up really tightly. And think about fishing line. If you have a long strand of fishing line, and you're going to move it somewhere, and it's laying out all over the floor and all over the room, how are you going to move it? Let's say I have to take some fishing line to Miss Mullis's room. I don't just grab the end of the fishing line and start walking to Miss Mullis's room, and I get in there and there's fishing line coming all the way back into my room. That's not a good way to move fishing line. A better way to move fishing line would be to reel it up. Get it all together on the reel, and then take the reel over there, right?
Right.
I'm a genius when it comes to moving fishing line. That's exactly what the cell does. And this shows the process by which the long strand of DNA is reeled up. You have that picture? Dang it. We're done. They got rid of it.
So that's prophase winding up.
Prophase is the winding up of the DNA into a chromosome. I got great video of it. The reel that the DNA winds around are actually proteins called histones. These little histone proteins, you can see the DNA is wound around them. And it's not like there's just one reel that the DNA is wound around. There's hundreds of reels, and the DNA winds around them, and then these little histone proteins kind of come together to make this condensed thing called a chromosome. And I have a beautiful video of it. It's one of my favorite videos. Check this out. It's a computer model. And you see that little line there? That little thing there? That's the DNA. And this blue blob there, that's a histone protein. And it's hard to tell here, but the DNA kind of loops around it a couple times, and then goes up, loops around a couple times, and then goes up, loops around a couple times, and then goes up. So there's many histone proteins. So watch how this DNA coils up. Everybody watching? You don't want to miss this. Took scientists years to figure this out.
That's so.
That's a chromosome.
We watched it again.
Absolutely. So these histone proteins, they all come together in a little circular arrangement. And the camera backs away a little bit. You can see there's a lot of these circular arrangements, and they wind up even further. And there's a bunch of these winds. Of course, there are enzymes that are helping coil it up like that.
How long does it take?
To coil up these chromosomes? In a quickly dividing cell, prophase happens in maybe 40 minutes, where these chromosomes coil up, and then suddenly you can see them. And notice how they're shaped like an X. There's a reason for that. This piece right here has an exact copy of itself attached to it. This and that are exact copies. Identical. Yeah, it does look kind of like a butterfly. Because remember the DNA copied itself in the S phase? There's the copy. It's attached to the original. And so we call each piece, the whole thing is called a chromosome. But each piece is called a sister chromatid. These two things are called sister chromatids. So let me write these on the board here. I kept too many magnets. There's way too many magnets. That's from Family Guy. There's way too many ostriches. Anyone ever seen that one? Love Family Guy. Okay, so what was that?
Sister chromosome.
Okay, so you have a chromosome. A chromosome is coiled up DNA. Each little piece here is called a sister chromatid. And you can see the chromosomes come in different sizes. But each one of them has a sister chromatid, two sister chromatids, attached in the middle. The middle, where they attach together, has a name too. It's called the centromere. The unfortunate thing about all these words is that they all start or have C in them. And it gets confusing because there's about 4 more words with C in them. So this is what happens during prophase. The chromosomes coil up.
What's a centromere?
The centromere is the middle, right here, where the two sister chromatids are attached to one another. This is exciting, isn't it? The cell's going to divide.
It's a boy.
It's going to be an identical copy. So up here on the board I have 6 chromosomes. Up here on the board I have 6 chromosomes. There's actually 46 in a human cell. There'd be 46 of these. 92 sister chromatids. But the 2 sister chromatids are attached in the middle. So there's 46 chromosomes with 92 sister chromatids.
So when it does copy in S,
Yes,
is there still attached?
they're still attached together.
Okay.
They stay attached together in the middle. Question, anybody?
Is there ever a time when they break apart?
Yes, that's what's going to happen.
Wait, you said there's
Exciting.
64 chromosomes?
46 chromosomes.
46.
And each chromosome consists of 2 sister chromatids.
I thought you said 64 and then 92.
46 and 92. I never make math errors.
I think so.
One time I did make an error, but I was mistaken. It wasn't an error. You get it? Okay. So, the error was that I was mistaken that I made an error. I should put that one on the board. I don't think it's mine.
Where do you come up with those?
Just random thoughts. Okay. So that's what it actually looks like under the microscope. That's a real good microscope. That's a scanning electron microscope. Pretty picture. 10,000 times magnification.
Can you see them under the microscopes you have?
Yeah, we can see, and we're going to look at them. We're going to go in there and look at them under the microscopes, but they look like just tiny little black lines. You can barely see them. You're like, oh, that's not very exciting. This is exciting, but we don't have it in an electron microscope. You can see there's a protein right here called the kinetochore that's on either side of the centromere. And that kinetochore is going to be important. It's going to help the whole thing break apart. We'll talk about that in a minute. There's a table in your book. This is a little bit more extensive than the table in your book on 157, but it shows how many chromosomes different organisms have. Human cells have 46 chromosomes. Chimp cells have 48 chromosomes. Goldfish cells have 94 chromosomes. The southern adder's tongue fern has 1,320 chromosomes. That doesn't mean the southern adder's tongue fern is more complicated than a human. It means its chromosomes are smaller. They're real tiny chromosomes, and humans have bigger chromosomes.
Why are there different sizes of chromosomes?
That's just the way it evolved over billions of years. Some chromosomes are longer than others, and the reasons for evolution are complicated, and we don't know them all. But we'll talk about evolution in a later
Do we know
video.
there's a particular reason for the so many of the first?
I don't know.
Question, Charles?
Yes?
Is that why chimpanzees and humans are kind of close?
Yeah, well, chimpanzees and humans are pretty close, and they have a pretty close number of chromosomes. You usually see that in organisms that are pretty closely related.
Does this happen like really fast? Like this whole process?
The whole process of mitosis takes about an hour or two in a quickly dividing cell. And interphase might take 22, 23 hours in a quickly dividing cell. So a quickly dividing human cell can multiply about once a day. A quickly dividing...
Like a bunch of them doing it.
Yeah, there's millions of them all doing it at once, so you get a lot of division. Yeah.
No, what's the do you know what the lifespan for like the average cell is?
It depends on the cell. A brain cell will live your whole life. Skin cells die pretty quickly, about 2 weeks. So it depends on the cell.
But do they start like dividing almost as soon as they're like?
So your skin cells do. They're just almost constantly dividing. Okay, so here's what happens. There's other things that happen in prophase. The chromosomes coil up, and then we got to get rid of the nuclear membrane that surrounds them. So you'll see the nuclear membrane dissolve away. And the little centrioles, the centrioles that existed outside the nucleus, they start moving. And they're going to move to opposite sides of the cell.
Is this still prophase?
This is all prophase.
What's the really big thing around? Is that the cell?
This is the whole, so this is one cell.
Okay.
And this was the nuclear membrane, and it breaks up and dissolves. So see, we're going to move these things around the cell, and we can't have the nuclear membrane there in the way. So enzymes will eat up the nuclear membrane, and it just disappears. And then these centrioles start shooting out microtubules called spindle fibers. Come shooting out. And what they are, they're little ropes. And the spindle fibers are going to attach to the chromosomes. The spindle fibers attach to those kinetochores that I showed you. The kinetochores capture them. And the little kinetochores have a motor molecule on them that can walk along the microtubules, you see, and carry the chromosome with it. So it allows for movement of the chromosome. So all that happens, and every chromosome will be captured. These fibers are going everywhere. So all of that's prophase.
So, what are the little yellow things called again? Centrioles?
These little things are called centrioles.
They just move from one side of the cell?
Yeah, they're going to actually move. They move across the cell, and you can see them moving here. And eventually they reach opposite sides of the cell. And they shoot the fibers out. And when the chromosome captures the fiber, we're actually in a phase that's kind of between prophase and metaphase. It's called prometaphase. When the chromosome actually captures the fiber. This is hard to see on these still pictures, so I have videos that show this a little bit better. This is a video of the chromosome coiling up. It's a great video here.
Here's a nucleus in early prophase. This time-lapse movie shows the chromatin condensing to form visible chromosomes. Here's a nucleus in early prophase. This time-lapse movie shows the chromatin condensing to form visible chromosomes.
See, you can see them now. Isn't that cool? The chromatin, it's called chromatin when it's just laying out here. That's another C word you need to know. It's called chromatin when the DNA is just laying out in the nucleus. It's called chromatin. And then it's called chromosome when it coils up so we can move it. Bam. And this is an artist version of what's going on, but it's also informative.
During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle.
Oh gosh, I got another C word. Those little things there, they're called centrioles. Did any of y'all remember when we studied the cell, you only find them in animal cells?
Yes.
Y'all remember that? The location where those centrioles are is called the centrosome. The centrosome is just basically a location where spindle fibers, these little fibers here, they're called spindle fibers, come shooting out of the centrosome. Animal cells have centrioles in their centrosome. Plant cells don't have centrioles in their centrosome. So that's two more words there. Centrioles and centrosome.
Centrosomes are in plant or animal.
Both plant and animals have these centrosomes. These areas here where these fibers come shooting out. They're called spindle fibers. They're just microtubules, part of the cytoskeleton. And it comes shooting out of the centrosome. An animal cell has centrioles in the centrosome and a plant cell doesn't. A plant cell divides just as easily as an animal cell divides. So scientists don't know what the centrioles do. They just know that they're there in animal cells. Savannah, you getting all this? This is not easy stuff. Continuing with prophase, or actually prometaphase here, we're going to lose the nucleus.
During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole.
So in prometaphase, these spindle fibers are captured by the kinetochores. So we call a microtubule that is captured a kinetochore microtubule. Because it's captured by the kinetochore. That little black dot is called the kinetochore. These two aren't captured by anything, so they're non-kinetochore microtubules. And what they're going to do, these two fibers are going to push against one another, and that stretches the cell apart.
So the microtubules are the little strands?
Yeah, these are microtubule strands. Do you remember the cytoskeleton we studied was made of microtubules and microfilaments? It's just like a tube of proteins.
There's always 9 of them.
9? I don't know where you got that. I never said that.
It says in the video.
No, I think they said non-kinetochore microtubules.
I thought it said 9 being took on the other 9 or something like that.
I don't think so.
Well, remember that's
In the book
that's
there's
the
12.
During
formation.
prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole.
It's
No, look.
non-kinetochore.
Look at this word. Non-kinetochore.
There's 12.
No, there's not 12 yet.
There's no number. It doesn't matter. I'm saying
There's no number. There's millions of them.
drawn there, but
Just put over 9,000.
It's okay. I mean, if I knew the number, I'd whatever. There's a bunch of them. Okay, then you get to metaphase. Metaphase is pretty simple. The little motor molecules move the chromosomes to the middle. So they get all lined up here in the middle.
What's his face? Metaphase?
Metaphase. Metaphase, middle. They both start with M. That's pretty easy. Metaphase, they line up in the middle.
I'm the worst filmer. Sorry. Keep going.
That's okay. Nobody's going to watch anyway. So, I'm sorry. You want to see video footage of metaphase? It's not very exciting.
Same guy.
At metaphase, the mitotic spindle is fully formed, and the kinetochore microtubules move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles.
So now they're lined up in the middle. Boop, boop, boop. Line up, line up. You can see this under a microscope. Our microscope, you'll see it. They're all lined up right in the middle. And you go, oh, there's a nice little line. Next phase, anaphase. In anaphase, the chromosomes separate at the centromere and start moving along the microtubules to opposite sides. So this is the part you were waiting for, Jordan. They break apart. I never had a problem remembering anaphase because in college I went out with a girl named Anna and we broke apart and moved to opposite sides of campus. And she was kind of a stalker, so she comes in. Yeah, question?
I thought you said the copies were like right above each other. So like the top one was like the bottom one's like the exact copy of the top one.
No.
Okay.
This and that are exact copies.
Okay. I thought you meant like.
And so the reason for all this, and that's good that you're thinking about the copies, the reason for all of this is because each side is going to get a copy. So that we've copied the DNA exactly, and now each new cell gets a copy. And so that's the whole reason for copying and then distributing. Other questions? Yes?
What's on the metaphase plate?
The metaphase plate is just this line in the middle when they were all lined up. Want to see anaphase video? So here's actual video footage.
During anaphase, the chromatids separate and are pulled to opposite poles.
Crazy.
Separate.
That really is crazy.
Separate. Separate. That's some good microscope footage, isn't
That
it?
is.
That's done with a phase contrast microscope.
How do you videotape a microscope?
You put a camera where your eye would be on the lens. You can do it with your iPhone in there. I'm serious. Next time we're looking at something, stick your iPhone camera right up to the hole. You can film whatever's on there. Stephanie was doing it. And, hey, you're pretty good at it, aren't you, Stephanie?
Yeah.
She got some real good pictures in marine bio. It takes a little, it's not the easiest thing to do, but it can be done.
Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. The kinetochore microtubule is shortened, pulling the chromosomes to opposite poles. At the same time, non-kinetochore microtubules lengthen, pushing the two poles farther apart and elongating the cell.
So I want you to notice in this video, the cell's kind of getting longer here. As well as the chromosomes moving to opposite sides, the cell's stretching. So this thing's going to break in two pretty soon. That's what we want. And now we got chromosomes, a set of chromosomes on each side. We call these are no longer called sister chromatids after they break up. They're called daughter chromosomes. So when they're together, this is one chromosome, and these are sister chromatids. Once they break apart, they're each called daughter chromosomes. Want to put that up as a C word?
Starts with a G.
Daughter chromosomes.
So sister chromatids and daughter chromosomes.
Sister chromatids become daughter chromosomes. Okay, the big finish is telophase. Telophase, these chromosomes arrive at opposite sides, and the spindle fibers all disappear. Once they've arrived, the spindle fibers will disappear.
What are spindle? How do they use this?
The spindle fibers were the little fibers, the little microtubules that were coming out that they were crawling along. Somebody else? Somebody else said how to something.
How do they just disappear?
Oh, there are little enzymes that break them apart. I actually showed you a video way back in chapter 4 of a microtubule kind of breaking apart. I don't know if you remember it. It's very cool.
Oh yeah, look at that, yeah.
And a new nuclear membrane forms around these chromosomes. And that'll happen on both sides. The spindle fibers will disappear, and a new membrane forms. And that's telophase. Since the chromosomes have arrived, they don't need to be all coiled up anymore. So they're going to uncoil and go back to the spread out chromatin that we started with. And now you'll have two new nuclei. And that's telophase. Telophase is when they arrive and the new nuclear membrane forms, and then they kind of uncoil. The cell itself then pinches in and splits in two. And now we got two cells. Question?
What happens to everything outside the nucleus, like the mitochondria and stuff?
They're just kind of floating around there, and if it was on this side, it ends up in that cell, and if it was on that side, it ends up in that cell.
So it's like an uneven amount?
Yeah, it could be uneven, yeah. Uh-huh.
What do you mean by
John.
Pinching in, imagine I took this belt, and I tightened it and tightened it and tightened it, I'd suddenly pinch myself into two halves. That's almost exactly what the cell does. It's got a little belt that tightens and pinches it off.
Then 0 say to 8. Nice belt.
During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil, and nucleoli appear in the two new nuclei.
Wait, watch it.
Meanwhile, cytokinesis begins, splitting the cytoplasm, separating the two daughter cells.